So hello everyone, welcome to our distinguished lecture series, which is taking place at Max Planck Institute for the Science of Light in Erlangen, and it's the most prominent lectures that we have at house. So just to remind you, this lecture is recorded and is broadcasted online via Zoom, and it will be uploaded in YouTube. So if you would like to go back to the lecture, you have the chance to do it in YouTube. So it's our great pleasure to have Professor Petra Schwille here today at our institute. She's a director at Max Planck Institute for Biochemistry in Martinsried in Munich. And she's an honorary professor at Ludwig Maximilian University of Munich. She's very well recognized for her ground-laying work on fluorescence cross-correlation spectroscopy and two-photon cross-correlation spectroscopy, and her current research is more focused on synthetic biology and bottom-up approaches to building an artificial cell, that we are going to hear more about it. So just a quick review of her academic path for young scientists in the room. She obtained her diploma in physics from the University of Göttingen in 1993. And then in 1996, she got her PhD from TU Braunschweig on the topic of fluorescence cross-correlation spectroscopy. Then in 1997, she left to the US, to the Cornell University for a duration of the postdoc, and she was back to Germany in 1999 as a research group leader at Max Planck Institute of Biochemistry. Only after 3 years of her position, she got a call of a professorship from TU Dresden, and then of course Munich wanted her back, so at 2012 she was back as a director at the Max Planck Institute of the Biochemistry. So many awards, just to summarize briefly. In 2001, she got the Lecturer Award by the German Chemical Industry Fund. In 2004, she got the prize of Philip Morris Research. In 2010, she got the Leibniz Prize of the DFG, which is very prestigious. In 2011, Braunschweig Research Prize. In 2013, Suffrage Science Award by London. In 2018, Bavarian Maximilian Order for Science and Art. And in 2021, Otto Warburg Medal. So we are very much looking forward to your talk. Thank you.
Yeah, thank you very much. I feel very flattered that you cited all these prizes. Yeah, so it just occurred to me that I'm giving a lecture in the Max Planck Institute for the Physics of Light. And I'm not talking about fluorescence correlation spectroscopy, and this is actually a big mistake, but I just like to talk to you. Actually, there's a little bit of fluorescence correlation spectroscopy towards the end of my talk. But I wanted to bring something else to you, which I'm very excited about and very concerned with since the last 10 years, basically. And that to end up in this field of research from, you know, being a very solid single molecule optics person, I would like to motivate to you by maybe some of you who are also application people and trying to bring your fantastic optical techniques into the real world. I think that is happening here in Erlangen all the time. The real world meaning some bodies or some organisms that you would like to measure in. I guess you all know this problem, that the fantastic signals that you usually collect, or the fantastic data that you usually produce if you do something in the test tube or on an inorganic substance, will suddenly, you know, become much more noisy, much less beautiful as soon as you enter a biological system. And this is because biology is super complicated. And because every biological organism in the world, even the smallest bacterial cell, is horribly complex in the way that you have an enormous number of parameters in there that you cannot possibly keep under control. So even the smallest organism has about 1,000 genes that you can find on Earth. And that means that if you have your fantastic technique, like single molecule optics, you're measuring something, You get very ugly signals. You measure something on one day and you measure something on the next day, and they don't really correspond very well. And that's because the system is not very well controlled. I mean, the more you zoom out, the more rough your technology gets. I just had a fantastic, where are you here, introduction in endoscope, and of course there you can pretty much rely on getting data that more or less is comparable from one day to the other. But as soon as you're looking at single molecules, things get very messy. So I basically like to have a very controlled system, and so I thought, well, which biological system is out there that I can control, or understand fully? None. So what do we do then? Well, maybe we have to build one. And what is a biological system? A biological system is, by definition, and this definition comes actually from the 19th century, a cell. A cell is the smallest possible biological system. That's a definition that people at some point made, and that still is valid today, even though there has been a virus recently, which we almost considered as a biological entity in the way we discussed it in the press. And there is some debate whether viruses are, living. But by definition, not yet. Not yet meaning the definition is not yet there. Everything that needs to be alive has to have a metabolism, and this is only possible in a cell. There's another thing with cell theory, and that is much more worrisome in a way. And that is that every cell comes from another cell. I mean, this is a great accomplishment, or was a great accomplishment at the time it was actually stated, which was, as I said, the 1900s or the 1800s in American or English, terms. Because at that time, people still believed that life could originate from mud, right, or some, dirty stuff. So people thought that, well, somehow there's, some spark going into mud and then life's coming out from the mud. Well, that is not what happens. And that was something that Pasteur and all these people here showed. If life is coming out of mud, it was already in. So there's no spontaneous generation of any living organism in our world today. It's bad news for us, because this simple thing that I would like to have, it's definitely not happening from, scratch. But at some point, it must have been there, right? There must have been a cell long, long ago, somewhere in the universe, maybe on Earth, maybe somewhere else. That was the very first cell that all cells came from. And somehow this cell must have been assembled by things that were not alive. And because that happened once, it must be possible that it can happen again. Just under which conditions? What do you have to bring together in order to make it work? So this big old question, how does biology come from chemistry? Very important, not answered. We would like to approach it experimentally. And as I said, our motivation is coming from physics as a background. We all know the beauty of the hydrogen atom, a system that you can basically understand from, fully or more or less. A real physicist would never agree to this, but at least we basically claim it would. A simple system, a unit, the minimal unit. We would like to have a minimal unit of a living system. So, and how do we approach that? Do I have actually a pointer? Well, I can do like this. How do we approach it? There are two ways of approaching a minimal system. First of all, you can go the long and winding road from an existing organism and taking off complexity. That's the approach that Craig Venter is, for example, very well known for. So it's called minimal genome approach, where you basically say, okay, we do not believe that all the genes that are in an organism are actually necessary for the organism to survive. So we take, of course not by chance, but we take some of the genes out and see when will the organism still, or how long will it still be able to still survive. And so the success story of this approach is already a couple years old, where they actually managed to take out from a very small organism, Mycoplasma, they took out about half of the genes from 1,000 genes that this organism had. They took out about 500, of course, as I said, not randomly, but very well, rationalized. And the thing was still alive, so it could still, regenerate. It was very lousy. It was much less well-performing as the original organism, but it was thriving. The problem is with this approach that from these 500 about genes that are still in, about 150 are completely unknown. They don't know what they're good for. They just couldn't take them out. That's not really what I mean with we have it under control, right? This is, well, it is working, and it might be a very good approach if you just want to reduce, the metabolic load of an organism, which is what the biotechnologists want to do. But in terms of understanding what you have, it's not really so helpful. So our approach is to build it up from the scratch, right? To basically say, okay, let's take the Lego approach. Let's go from the level of modules, and I'm deliberately not talking about molecules or even atoms, which we in principle should do, because the way is very long from an atom to a cell. It's still very long from a module, and I will explain what I mean with module, to a cell, but at least it's kind of conceivable. And the other reason why we do not start with atoms is that we are not chemists, but physicists. So the idea is assembling with modules, with a, basically like a Lego approach with modules that, as you can already see, are relatively complicated molecules, namely proteins. We are building with proteins, and I'm coming to the problem of this approach, but take it at the moment for, this is the modules that we can understand as functional, right? So if an engineer wants to build something, you better know your functions of the modules that you're putting together. So we are taking functional modules and we put them together, and hopefully start from, a simple system to a system that has the complexity that you could call it alive. Now I'm coming to the conceptual problem with building with proteins as modules. As I said, originally you would like to have something simple. For sure, proteins are not at all simple, and also they are not fundamental. They presumably arrived relatively late in the origin of real life on Earth. We do not know that, but this is at the moment the hypothesis that first there was RNA, and then at some point there came proteins. Much more worrisome than the conceptual challenge, because I still hope that as long as you understand how function emerges from simple function, you're on a good track. But we also have a technical challenge, and that is something that my people, the protein, the people who are working in the lab every day with the proteins, have much more as a headache. They work fantastically well in the organism, but they do not work fantastically well if you take it out of the organism. Some do, and most don't. So it's a technical challenge to get them actually to do what they're supposed to do if they don't have their native environment. And of course they don't, because we want to keep it simple. And then there's an academic challenge, which I would not like to, go too much into detail, because it's mostly to the students who are studying biology or life sciences, because usually the category of success or category of excellence in the life sciences is always physiological relevance. So if you have a study which claims to be physiologically relevant, that you can basically learn something about a real disease, it is usually very well received in high impact journals. Studies on purified proteins unfortunately suffer from a lack of physiological relevance, unless it's a very well-known protein that is clearly a tumor marker or something like that. So this is a little bit of a problem. I think we are here at Max Planck, so a lot of fundamental researchers, the fundamental researchers have this privilege of not so much worrying about applications and impact in the citation world. Okay, but how do we actually go about this problem? What do we want to build? How do we start? Where do we want to go? And here, we have to at some point define what we understand as a living system. everybody more or less knows if a cell is lying under the microscope, if this is a cell, but a simple system, when would we call it alive? What particular features does this system have to fulfill or to show before you actually are convinced that it is living? Well, a very famous physicist, Schrödinger, he wrote a fantastic little book, which is not very much liked by the biologists because it's a lot of stuff in which is then was that basically proven wrong in the 50s when the DNA structure was revealed. But he said something very, very fundamental and very true about living systems, namely that living systems are able to create order out of disorder, not only create it, but also maintain it on the expense of energy, which is flowing through the system, energy being the sun energy, or light energy, or also chemical energy. And the maintenance of this patterns, of this structure, of this order, is something that not only living systems can accomplish, but living systems are particularly good at it. So what we need is from our system is that it's able to form patterns, that it's able to self-organize upon energy consumption. And then there's this big, big part, which is basically determining how this order is maintained by energy input, which is called metabolism. And this is the world of biologists and chemists, and I admit that I have very little knowledge. This is all the, carbon cycles, these horrible things that you look in textbooks and you get completely scared away from it. So lots of molecules doing lots of chemical reactions. So don't mess with this one. We take this as granted. But look at something that a living system can also do, and which is much more appealing for a physicist, namely they can divide and multiply. And that's a tangible goal for somebody who, is interested in physical processes, because it's a very physical process. A system that is able to make the mechanical transformation, basically assembling somehow a machinery that just divides it either in half or in other daughters. This is something that you can approach with physical methods. There's force needed to do that. There's, distribution of molecules needed. All of this is something we can easily see. So this is something that we would like to accomplish. One only of these many, many features that the living systems needs to fulfill, but for sure needs to fulfill. It needs to divide and proliferate. How do we build a system that can do that? That is our question. Now, a long time ago, when I was still in Dresden, I'm not even sure whether Jochen was already there, we basically stepped upon a system that beautifully combined this ability of pattern formation with the fact of, with the feature of cell division. And this system is the bacterial min protein system. And this is a very interesting protein system which oscillates, and by this forms beautiful patterns. In this case, it's basically a volume oscillation. You have more molecules on one side than the other between the two poles of E. coli bacterial cells. And what does it have to do with cell division? This oscillation is a spatial cue for the machinery that is supposed to divide the cell. E. coli cells need to divide in the middle. They have to distribute their genome in the two halves and basically then assemble something like a ring structure, which by forces we do still not understand fully, is able to basically make a scission movement and divide the two halves of the daughter cells. And the positioning, as I said, of this ring into the middle at the very geometric center is performed by these oscillations. These proteins that you see oscillating, which are of course fluorescently labeled, they prohibit the assembly of the ring in the pole region. So they basically wash this ring machinery away from the poles, so it can only assemble in the middle. So since it is very clear to see how this machinery functions, how the emergence of this machinery is actually established, and this is a phenomenon which is driven by ATP hydrolysis, so a very clear metabolic process. This was something as a starting point that we wanted to show in the lab. And I had a very brilliant PhD student, Martin Lohse, who accomplished this purification of this machinery from the bacterial cell into a test tube. So what we basically have in this test tube is a membrane that mimics the inner side of the bacteria where the proteins bind to. Then we have just a buffer solution with ATP as the energy source and the proteins. There are 2 proteins, and that's very important, and I'm talking about the mechanism later. One protein is binding the membrane, and the other one is taking it away from the membrane. And if you, oh, I'm sorry, that was a bit too fast. Let's go back. If you take that back, if you basically start looking at these proteins in this very reduced system, only the 2 proteins and buffer solution and the energy, you can see the self-organization very beautifully. And that, of course, that was a big breakthrough, but of course this is still not something that you can use as it is in a synthetic cell. So first of all, you have to get somehow this fundamentally this beautiful pattern forming system in a compartment that could act as a cell. This is still, just a cover slide covered with a membrane. And it was actually Katja Zieske, who is a group leader here, who accomplished the first step towards enclosing this pattern-forming system in a cell-like compartment. At that time, it was not a vesicle because it is very complicated to keep a lipid vesicle, which is basically a little bit like a soap bubble, very delicate object, and the proteins happy. So the trick that we did at that time was basically to just cut little grooves in a microfluidic chip, in this case PDMS, and basically coat it with membrane so that the proteins always see the membrane on it, like a bathtub which is lined out with membrane. And if you fill your system in there, you can actually beautifully see how the system does exactly the same as it does in the bacterial cell. It oscillates between the 2 poles, and it does that without the need of any other factor than the 2 proteins and ATP. So this is a simple system, and it works. And now the next question is, does it also, as claimed for the bacterial cell, does it also position this ring-forming machinery that is supposed to orient the division? Yes, it does. So what you see in this red and green movie is that these are the min waves on a flat surface, and the red little things are the filaments that are supposed to form these rings. They're made of a cytoskeletal protein, FDSZ, which is usually assembling in ring-like structures. Of course, not in this geometry, in a flat geometry, but here you can see that the min waves are basically chasing them away. And if you look it all up in a compartment, you can appreciate, so first of all, and that was also something very nice that Katja saw, that the length of the compartment basically changed, of course, the period of the oscillation. So the speed of the wave was always the same if you had the same concentration. So the period was very clearly dependent on the length, and that makes a lot of sense because the cell can only divide at a certain length to width ratio. So it should not divide when it is still small. It's growing in only one direction, and as long as it's still small, it should not grow. Only if it has reached a certain length and the oscillation has then a certain period is the division supposed to happen, and that is about the length of 4.5 length to width ratio. And what you see very nicely here is that the oscillation splits up into daughter oscillations in these, mimicry of a cell. Although, of course, here nothing can divide because it's just a bathtub. But what you also can see very beautifully is that what was supposed to happen happens, namely that the concentration of these inhibitory green proteins that are supposed to inhibit the formation of the ring in the middle is indeed smallest, and it is that is the reason why, oh no, I lost my cursor. No, here it is again. And that's the reason why these filamentous molecules, they can orient only in the middle. So here you can see how this system spatially self-organizes and orients its modules autonomously without any other cue than the chemistry which is basically underlying it. Of course, here we do not see a full ring because the system is still open. So it's in an open bathtub, you cannot form a ring around. So from here on, we did a lot of characterization of our system because it's, of course, a very interesting system. And we had a collaboration with a theory group in Munich, a group of Erwin Frey. And we basically wondered about the features of the proteins and how exactly they form this pattern. So it appears that there are two factors that are relevant for this system to, create this emergent behavior. One is the binding of the membrane, because the molecules need to have the membrane in order to form these beautiful patterns. And the other one is the ATP hydrolysis rate. And we found that the membrane affinity is a super important parameter whether patterns are formed or not. So we could find that if this is the native wild-type molecule, if you change only one amino acid in the membrane targeting sequence of this protein, you still have patterns and you still have dynamics, but the oscillations from pole to pole are completely lost. And what we also found interesting is that you can basically make one of the molecules you can take it completely off the membrane. So it only one of them can still bind the membrane, the other one cannot any longer. You still get patterns, but the oscillations that you get in your compartment are completely opposite to what you have in vivo, where you have a pole to pole. Now here you have a top to bottom oscillation. This cell would, of course, not be able to divide. But in vitro, you can beautifully see that the system is still able to self-organize. And we go even further. So the system of two proteins, so they are not very complicated proteins compared to many other proteins we have. They are relatively small, but they have complexity in them that we did not understand. For example, one of the proteins has a conformational switch. So one binds this one here is the more relevant one. This is annoying. Here. This is the more relevant one. This is an ATPase. This is the one that always binds the membrane. And the other one is basically taking it off the membrane. But in order to be active to take it off the membrane, it first has to undergo a conformational change from a closed to an open state. And our theory partners basically said, well, we don't understand that. In principle, this should also work if it is always on, it's always active, it's always open. So we cloned a version that is always open and found that the system still works. The difference is that the system, this is the simplified version where you always have an open molecule, and this is the wild-type molecule. And what you can see, you get patterns for both types, but for the simplified one, the stability against concentration changes is much reduced. So if we vary the concentration ratio between the two molecules, in the case of the simplified version, relatively easily we lose the ability for self-organization. So here you already, so it's this blue background. Here you can already see, I don't know what this is. I know, I need to stay in the left side of the okay, now I understand my computer. So here you can see that after only a short concentration change of the ratio, the ability to self-organize is lost, whereas in the wild type, with the higher complexity, with this additional switch, you can see that it's much more tolerant against concentration changes. And that's actually what nature is good for, right? It's known for being resilient. It's good. It's known for being, not super critical. A bacteria that could not control its concentration range would die right away. This is the reason why nature is so complicated. But of course, it's a nightmare for us. But now we understand what we can take out to understand still the system, and that's good news. We even went further and asked, could we reduce the complexity even further? Because this molecule that we are working with, the smaller one of the two proteins, it consists of 4 motifs. One is this, this is the active catalytic helix, which is talking to the other molecule. So this, of course, we cannot take away. But besides that, we have a dimerization module. We have this conformational switch that I talked about, and I also talked about the membrane targeting. So we saw we can take away the membrane binding, we can take away the conformational switch, can we also take this motif away and reduce the protein to a super, super simple helix? Well, apparently we cannot do that. So we need at least either a membrane attachment attached to the a membrane motif attached to the helix, or a dimerizer attached to the helix. But we can either or. And what is even more important, we do not have to take what nature provided, the native membrane attachment or the native dimerizer. We can take anything that dimerizes the helix. And now the engineering aspect comes in. If you can just dimerize it by anything, why don't we dimerize it by something very simple, for example, a DNA? So one of my students, she just fused the little catalytic helix to a DNA single strand and offered, you know, the complementary strand to the other helix, and could show that this, hybrid molecule can still create the patterns that the protein can. So we have now done something very important. We have now basically gotten behind the protein level. We went from the protein level to the functionality level. And that's very important, because if we want to create something from scratch, it would be a bit complicated to do it all on proteins. But since we have now understood what is needed, namely just a dimerizing motif attached to a catalytic helix, we are a fast step into, creating something that is much more simple than a living system. Of course, this is only the tip of the iceberg, right? We have only just replaced one already relatively small protein by a very simple molecule, but it's possible, and that's the most important message. Okay, but let's go to the maybe a bit more interesting molecule, the one that is supposed to actually do the scission in cell division. Well, the problem is, as I said, it's still not fully clear how E. coli cells actually execute the division. It's clear that they do it, but how many factors are needed to do this, cutting not only of the membrane, but also of the stiff cell wall? There's a huge machinery involved in the bacterial cell, but it's very clear that it all kind of orchestrates around this scaffold, which is a bacterial cytoskeleton molecule, FDSZ, which is a homologue of the tubulin, which is making these big microtubules in our eukaryotic cells. What the peculiar thing about this scaffold protein is that it self-assembles in filaments that are curved, that are intrinsically curved. So if you purify the molecule and put it on a membrane, you can see that it forms little rings. So this is much, much larger. So this is about to scale here. This is a micron in diameter, and this about 50 microns in diameter. So it always forms rings about the size of a bacterial cell. And if you look at these rings very closely, you can actually see that they're not only beautiful and round, but they're also dynamic. So they basically treadmill. And you might recall, if you have if you had biology classes, that microtubules do that. Microtubules treadmill, so they grow on one side and they shrink on the other one. And this is exactly what the FDSZ is also doing, with the difference that it's not directional. It's directional, but it's always going in circles. And we found that it always, if you look at the wild type, it always goes in a clockwise direction. And we found that this is because of a chirality of the molecules. So they have basically three axes of symmetry. One is the directionality of the assembly and disassembly. One is the curvature, and one is the membrane attachment. And they all three of them are kind of orthogonal to each other. And you flip the membrane attachment to the other side, the N-terminus of the molecule, and then you can appreciate, not quite as beautifully, but you can see that the rotation is reverted into anti-clockwise. However, what we originally hoped to see, and this is all energy-driven, so also treadmilling is, of course, something that is dependent on energy, in this case CTP. GTP, excuse me, GTP. If you look at these, in principle, you would expect if you have a filament that is consuming energy, you would actually assume that it's somehow contracting, because this is supposed to happen if the cell divides. But there's no contraction whatsoever. They're always keeping their diameter. They never shrink in diameter. And of course, this is a bit of a disappointment, because it means we cannot just rely on the shrinking of, of this ring to divide a vesicle. But what we see is that if we reconstitute these rings in a tube, of course here they are on a flat surface, which is completely different from a bacterial cell, where they are basically around the circumference of a tube. So in order to create membrane tubes that are comparable to the bacterial size, we pulled with an optical tweezer from these FDSZ decorated vesicles. So these are just membrane vesicles, very soft. So we pulled a tube, and if you do that with an osmotically deflated vesicle, you can get tubes that are about 0.5 micron, or a bit less maybe in diameter, smaller than a bacterial cell, but about the size. And in this case, what we see is that our FDSZ enters the tube, and now, because we have broken the symmetry, they are not lying only on one side, they can basically now attach in both directions. We have something like a squeezing of the rings. So some of the rings are going in this direction and the others are going in the other direction. And by this, you're actually creating a force which will deform our membrane tube almost like a spring. So what we show here is not quite what we would like to have. We do not see that the tube is kind of reduced in diameter, which would be ideal. But what we see is it is deformed, and it is deformed in that way. And who knows, maybe this is the original engineering. Of course, this is something that we still need to, confirm. But maybe this is the original mode of membrane scission, that it's not kind of squeezed in this direction, but actually, maybe you recall this art with balloons, right? Where you have tube-like balloons, and if you turn them, then you can actually see how their little necks created. Maybe this is the mode how originally the molecule that were designed like this was supposed to do a membrane scission. Originally, I say, because a cell is no longer, especially not the bacterial cell, just a membrane tube. It's very strongly connected to a sugar layer, and you need a lot of enzymes to also build the sugar layer. So the complexity in the real biological cell, in the real biological bacteria, is so high we would not want to rebuild. But there might have been a cell before that, before the invention of a sugar layer, and that might have been split in a system, with a mechanism like this. Okay, to cut a long story short, what we originally wanted to do for a long, long time finally worked 2 years ago, or 1.5 years ago, where we basically got the oscillating system, the min system, where you could see in these nice bathtubs that oriented the ring structure in the middle of the bathtub. We could finally got to work in real membrane vesicles, and that was a huge accomplishment in terms of biochemical problem solving, also with regard to microfluidics, to fill the vesicles, keep the membrane stable. And it was 2 excellent people, Adrian Merino, biochemist, and Shunshi Koyama, bioengineer, who basically got that to work finally. So what you see here is the FDSZ filamentous system. In the absence of the min proteins, you can see that these filaments decorate the vesicles relatively isotropically. And now if the min proteins start to oscillate, you can see that these isotropic filaments will basically all be driven in the middle, basically there where the oscillation drives them to. And then you can see how one single ring, more or less, is assembled. And this is very beautiful, but what we did not see here, because it was a relatively artificial system, was that there was any deformation by the membrane. Of course, as you appreciate here, the bacterial cell is not a sphere, and the sphere is the least appealing object to divide in two halves, right? So in principle, if you want to divide something, you first want to elongate it because the volume to surface ratio is really optimized in the sphere, right? But if we now take a little bit more biology in the system, and oops, sorry about that. Oh, I do. No. It needs to start. Why doesn't it start? Here. What we did here was to basically not only put in purified proteins, but produce the proteins already in the vesicle, which has the advantage that the timing of what protein comes when can be better orchestrated. And if we do that, it's still the same amount of modules, still only the 2 positioning molecules or 3 positioning molecules and the filament forming molecules that are supposed to form a ring. If you have the timing right and the membrane basically not completely swollen, then you can see very beautifully how not only this isotropic, filament structure assembles into a ring, and how this assembly in reverse orients the oscillation from pole to pole. But you can also see how the vesicle gets a neck here. Of course, it's not a very beautiful neck. It's still very, very small. So the deformation, the aspect ratio that we get is still, of course, very, very low. But we're getting there. And that's already very, optimistic. We are very optimistic about that. So this is as far as we have come with our, reconstitution of a cell division system. As I said, what we have here, it's not yet a division, but it's only 5 proteins. But there, the next steps here, who knows? We may want to elongate our system. We may want to basically combine it with something like an inner phase, which resembles the nucleoid of the, also, in every cell, there's not only the membrane and the division system, but it's also a DNA, a chromosome. And maybe the chromosome and the division of the chromosome has actually an active supportive role in the whole process. So we are at the moment exploring that. Or we are also exploring to engineer the ring a bit more so that the force can be improved. But I would like to show you something which I, put on my title, Life as a Matter of Function. Talked about function, and there's something about this approach with purified proteins which we found very peculiar, namely that the functions that we predicted, or that biology basically predicted for these proteins, we found, right? The orchestration of the pole-to-pole oscillation, and also the positioning of this ring. But we found other functions that these proteins have, which we did not expect and which biology does not describe. One of these functions is that the min proteins are not only positioning these ring-forming proteins, but they're actually positioning other proteins and even molecules that are not at all related with them. So if we put them on a membrane and put, this is very, just look at on the right side of the thing, because this is obvious. So they compete with other membrane binders. That's not so surprising. But what they do, if we have molecules on the membrane that can diffuse in the membrane, these are our min proteins, you can actually see how they drive these molecules out of the way. And there's no biological relation between the two. Just the presence will basically be, basically be driven like with a broomstick. It will basically be driven off the membrane, or it will basically be driven off a region in the membrane. And what is even more amazing is that you can make the molecules that can be driven away. No, it stops here. Why does it? Come on. Yeah. I should probably just, we made this little animation to exemplify what's going on here, what we believe is going on here. We think it is a diffusional ratchet. It's called diffusophoresis. This massive, carpet of proteins which are basically involved in this wave, they will not allow the membrane diffusing molecules to diffuse in backwards. They can only diffuse in one direction. Oops, I'm really sorry for this. I just want to play this again so that you can see it. So usually molecules can diffuse in all directions, obviously, and as soon as you have this, this big bulky wave of proteins, it will not be able to diffuse in two directions, and by this you can actually drive them. And that means that this transport effect, which is an energetically driven transport of membrane binders, is of course dependent on diffusion. So something that is not diffusing is not driven by them in proteins. But as long as the molecules are diffusing, you can actually drive them, and you can even drive them dependent on their size. So here we basically had two different cargos on the membrane, one with a lot of membrane anchors, so that would be a big bulky resistance against the min proteins, and one with only two membrane anchors. And you can see that the one with the many membrane anchors will be more efficiently transported away than the one with only two. So you can not only position, but you can also sort. So you can create patterns by this diffusophoresis of any molecule on your membrane, which is pretty cool. And the min proteins are not known for this in bacteria. What they are also not known for is that they can completely deform membranes. So here we basically put them, this was at the beginning where we accomplished the introduction into small membrane vesicles. We see not only pole-to-pole oscillations, because it's very hard to define a pole in a round vesicle, so you also have modes where they go around in circles, or where all the molecules go in the lumen, and then in the next instance they all go to the membrane. So it's basically a volume to surface oscillation. And if we have this, and then have an osmotically deflated vesicle, you can see that it's bouncing up and down. It's like a ball where we basically, you're tensioning periodically because the molecules go to the surface. And this can go even as far as making a full division. And there's absolutely no structural protein in there. There's only the oscillations. And just the, oscillating, tensioning of the membrane and loosening of the membrane will basically create these features. Another effect that is not to be seen in bacterial cells that have a stiff cell wall. But it's a function that is very clearly there. And even further, we put these molecules on a surface and put another vesicle on the surface. And so the min proteins can bind both to the supported membrane and the vesicle. And if we have something like that, the binding to the vesicle will basically squeeze it and deform it, and by this initiate a chemomechanical or a mechanical cycle that drives the vesicle forward. So this is a real a large-scale mechanical effect, which is basically driven by the oscillating reaction on the surface. All of these functions that are clear functions that can clearly be, in principle, harnessed for something, are not used, or at least not to our knowledge, used in the bacterial cell. So it means that proteins have not only the functions that they're known for, they can have many, many more functions. And it makes a lot of sense to study what functions they have, because if you want to assemble living systems from scratch with a minimal number of motifs, you better know about all these, unknown, hidden functions that proteins have, because that gives you the opportunity to build your system with much less parameters. This is basically the main take-home message. I have a couple of slides basically at the end of my presentation here that have some optics in it. The question is, do you need that or do you not? So what is the, are you tired, or do you want to have some optics? What is the usual time scale of the? Okay, so you get some optics from me. Because of course, you want to know what's going on here, right? So what are these molecules? What are they doing? How are they actually creating these amazing patterns? Well, since we're coming from the single molecule field, we, of course, first looked what is going on the single molecule level in these beautiful oscillations and patterns that we see. And if you look at, this is just this wave, zooming in this wave that you've seen earlier. If you just label single molecules and look at them, you can actually appreciate that they don't have a concept of moving directionally. So every single molecule in this wave is just binding to the membrane, diffusing around randomly and isotropically, and then dissociating from the membrane. So the effect of a coordinated movement is very similar to the effect in a football stadium if you have these Laola waves where people stand up and sit down and there's a wave going around, but the single person will not move, at least not to the left or the right. So this is what you have here. So the molecules are binding. They are basically, other molecules are more likely to bind where there's already one. This is a cooperative binding. But other than that, there's no directionality. Now the question is, can we somehow understand molecularly what the mechanism is? And this has actually been proven to be very complicated because you first want to understand what are the binding affinities here and how long are the molecules actually binding. And you might recall that I've done a lot of FCS in my career, so fluorescence correlation spectroscopy, where you basically take fluctuations of a fluorescent signal, and you're basically correlating these fluctuations and get time information about the fluctuations. And usually FCS is performed in a confocal setting. You have a confocal optical volume, and then molecules are diffusing in and out. And the fluctuation arises from the presence of the molecule in the volume. So you basically get diffusional oscillations or diffusional fluctuations by every single molecule. And if you correlate the signal and the fluctuation, you get the average time that a molecule spends in the volume element. This usually works on solution. But why should it not work on a surface? So we designed another way of doing correlation analysis, which does not mean we're taking the signal from the volume, from a confocal volume, but we're taking the signal from a whole plane of view. So we're basically averaging over the whole area where you have molecules coming and leaving, because this is the same fluctuation signal that you would have from a molecule diffusing in 3D. So what you do is you basically take this, you average it, and then you correlate it. And by this you can get something very beautiful. You can get basically correlation curves that tell you the average time that a molecule spends on the membrane. So what you see here are beautiful curves, but they're not beautiful curves from proteins, because we really never, it was because people, lost their interest, but the curves that we recorded were really from very boring molecules, namely from DNA binding to other DNA on a surface. So far we do still not have the data from the min oscillations, but it's basically the same concept. So you're taking labeled molecules, you're taking basically the average time they spend on the surface. You integrate and you correlate, and then you get the time scale just from the decay function of the correlation function. So here we basically established that with just different affinities, different overlaps of DNA single strengths that were on a surface and that were in solution. And the more overlap you have, the longer your DNA stays on the surface. Here you can very beautifully see that you can from 7 to 10 nucleotides, you're changing dramatically 2 orders of magnitude the affinity. So that was very beautiful, and I always, as I said, wanted to get that to the proteins, and it never really worked beautifully. And then we got introduced by a talk by Philippe Kukura. I guess a lot of people know him here, who basically introduced the mass photometry based on iSCAT, which is very prominent here. And I was basically, I thought, well, why would we actually need to have fluorescence? Why don't we just take, the scattering from iSCAT to do the same, basically finding out how long our molecules spend in our volume element. And that is what we did. And of course, it was very helpful that, there was this commercial instrument around from the Refine company, where we basically just took the same signal, but no longer a fluorescent signal, but now a scattering signal, and recorded our single molecule data. And it occurred that this is actually the perfect system to study proteins, because you could not only, and this is in complete difference to fluorescence, you could not only look at timescales, but you can also look at sizes. So we can not only look at how long does a molecule spend on a certain on a membrane, but we can also see how large it is. So we basically applied iSCAT, or mass photometry, if you will, to the question of how the min proteins bind to the membrane and how these waves actually grow. And I'm sparing you of all the details of the biochemistry behind. It's a very, very messy system. Still, we are not fully, behind the cooperativity of this, but it is absolutely the right technique to approach this biochemically. This I just wanted to show you, and now I'm in the end, and I very much thank you for your attention, and I also thank the many, many people who have contributed to the work. There are even more that I did not put on here, but I just concentrated on the most important ones. So again, thank you very much.
So thank you very much for the great talk, and now we can have questions. Any questions?
Thank you, beautiful. I have a question for the vesicles for the FTSZ rings, which, I mean, I'm just flabbergasted that you actually, in the end, managed to do this. And I know how hard it is to get such a multi-component system running in vesicles. So I think you correctly point out that maybe geometry might be a key factor, which is typically induced from inside and etc. But maybe we don't need to do that, as the cells do. What prevents you from putting these deflated into a microfluidic channel and then see if that actually changes? I mean, you must.
I constantly ask my people what prevents them from doing it. It seems to be very complicated. The problem is you have to make the vesicles first, and they usually make them by electroformation. They are still, I mean, you can make them by many ways, but if you want to keep all the proteins happy and all the lipids happy, then there's a certain parameter space that you can approach, and so far we have simply not accomplished it. Of course we want to do it, but it hasn't worked so far.
Stay tuned.
Okay, more questions. Yeah, vielleicht zuerst Jochen.
A very beautiful presentation, thank you very much. I want to follow up on that question. So if you deflate a vesicle, wouldn't that already give you more shape change that then the contractile ring could actually work on? And
yes, absolutely. The problem is it is not so easy. I mean, the deflation is also something that you can do to some extent. The vesicles where we have, it's just so far not working. This is how you, it is a pretty messy system, although it's, in terms of parameters, it's very complicated, it's very simplified. The vesicles that we create, where we can fill all our this is a pure reaction mixture, where I think people might know about it. It's basically a mixture where you can express proteins from, it's basically a soup. You put in the gene and it will express the protein. And this system does, for some reason, not 100% cooperate with the osmotic deflation.
you figure that out and you can actually pinch that off.
Yes.
The only thing that is missing is metabolism then?
Yes, well, the only thing, there are two big things missing. One is the metabolism and the other one is any kind of information unit.
Metabolism, so is that something that in principle you could put in, say 5 years, 10 years, all kinds of challenges eventually overcome? You put in metabolism, some sort of chemical cycle. How long would that then run? Is that then the next problem? How do you?
Absolutely, this is the next problem. Yes. I mean, of course, the way that cells operate, they have a super complicated metabolism, right? I mean, carbon cycle or the Krebs cycle, or this is this is totally crazy, right? You don't, I mean, in principle, you need a much simplified metabolism. You need something that is, in principle, metabolism means molecules are constantly made and destroyed. And ideally, the molecules that are doing the job need to be made and destroyed, and the energy needs to be made and destroyed. So it's still a long way to go. Yeah, but you know, I hope that the chemists among our synthetic biology community will have a solution to this. I certainly will not offer help there.
Yeah. I wanted to ask you here, like, I guess especially on this example with Escherichia coli, correct me if I'm wrong, but I understand it's basically that the system is super simple, so you basically have binding and unbinding, and you need ATP just for the energy. And here you have a mechanism to basically measure the binding energy. I mean,
Mm-hmm.
in theory, I mean, this is basically Turing pattern, right? I mean,
Yes, it is a Turing pattern. Yeah.
and is it, when you do if you measure the binding energy and you assume get these patterns, or is it also depend, or will the binding energy heavily depend on the local curvature?
In this case, not. Although, if you would have a very high curvature, I mean, we are not operating at this very high curvature. So what you see here as curvatures are very, very large, right? So it's basically the curvature of the Earth for a motorcycle or so. I mean, not quite as bad, but I mean, it is not a very strong curved system. Obviously, if you have a curvature in the size range of a protein, so if the lipid would be very curved, then there would be a different affinity to the membrane, and there you would definitely see it changed. But this is not the case here.
Petra. So I think the key word is apparently Turing pattern, but I would have thought that, so you have a mechanical wave, and could you describe what happens more in physical terms? In your language, as often you say proteins do this, of course the proteins don't do anything, right?
No.
Something happens that they end up doing, following a certain pattern.
Yeah.
So if you were to describe this mechanical wave, what is the cause of it? I mean, is some kind of an aggregation, some kind of a change in terms of surface tension or whatever happens? Could you break that down to?
Yes, I mean, we still don't know every single part of it. There's presumably some allosteric, transition going on, but in principle, what happens is you have this min-D molecule, the big molecule in this two protein system, which is an ATPase. So it as soon as ATP binds, it goes to the membrane. And when it is at the membrane, it cannot hydrolyze the ATP by itself. It needs the other little helper protein, and only then can it hydrolyze. But this other little helper protein will not come without a certain local concentration of min-D already on the membrane. So it is this is what biologists or biochemists always call recruit. I hate the word because there's nothing is recruited. I mean, you need a certain local concentration of molecules on the membrane in order for the effector molecule, which helps to hydrolyze ATP, to be, to come to the site, and then the whole thing basically breaks apart. So it's very likely a conformational change in the molecules that are on the surface that basically then fly all off the membrane. So in principle, it is a cooperative binding. So if there's already one min-D on the membrane, the ability for the next one to bind is higher. That we have checked. So it's cooperative binding, and it's a very highly cooperative unbinding. But besides this, we still do not understand the real, structural rearrangements in the molecules.
Okay, thanks for the nice talk. I had a question regarding the second to last slide you showed with the autocorrelation function.
Yes.
and on-off binding rates. So if I remember correctly, these on-off binding models, they usually, yeah, they go along like non-Brownian diffusion, like anomalous diffusion. And do you see this actually in the data here? And
there is no diffusion in. So this is on time scales where diffusion does not play a role. So usually diffusion, I mean, in the membrane is very slow compared to diffusion on, you know, in solution. This is all in turf. So we are measuring only, we're only measuring everything in the membrane. As soon as the molecules are in solution, we don't see them anymore. And since we are integrating over the whole field of view, a diffusion in the membrane doesn't play a role.
the temporal decay corresponds usually to a diffusion time, right?
If you have, if the diffusion changes your signal, but in this case, the diffusion would not change the signal. So let's just go back. You can easily see that. See, I mean, these guys are fixed, but even if they would diffuse, if you integrate over the whole field of view, it would not matter. I mean, a molecule, a point is a point, and since we're, as I said, we're integrating. We are taking all of the brightness here and integrate it, and it doesn't matter whether the spot is there or there or there, so diffusion would not to be seen here. But this is a very special kind of correlation in a diffusion in a volume. Clearly, you cannot discriminate against the diffusion, and you would see it.
Thank you.
Thank you very much for the talk. So while we are on these slides, you showed these measurements with the IceCat on mass spectrometry.
Yes, yes. Mm-hmm.
Then you said that it's very, I mean, it fits very well, and it's, I mean, I see that why it also fits very well. So the reason is that you can, based on the mass, you can identify who is where, right?
Exactly. I mean, this is this, you know, this mode that they have. I guess you're also using that. So you're basically subtracting. If a molecule has come and is already there, then they are subtracting the background. So you can see if the molecule moves, you can actually see how the black spot moves on the membrane. So you can see where the molecule is. You can track it over time, and you can get, you know, you can see that the size is growing. And that's something that we definitely very much like to see.
And then, do you happen to know what is the resolution of the mass that you can, the changes of the?
It's about the. I mean, our proteins are at the absolute limit, at least of that version of the refined instrument, which is, so how many kilodalton are our proteins now? I should know that. 30, 50 something. I don't know. That was basically, we were pushing the limits here very strongly, but it was possible to resolve the single units.
Thank you very much. Very impressive talks and videos. I wanted to ask something that you already mentioned very little bit about the information processing system. How would you build the information processing system in such artificial cells? Would you use DNA or something hand-created or different?
Well, so far we haven't really thought about that because we are battling with the division, but DNA is obviously a molecule that is very, you know, very much liked by everybody because it's very benign in order of to do experiments. So of course you would, I mean, something that we have not yet evaluated sufficiently, and I think what is at the moment being appreciated more and more is that DNA is not only an information encoding molecule, it also has a polymeric nature. It's in principle a negatively charged polymer, and it in a eukaryotic nucleus it's of course packaged, but in a bacterial cell DNA is in principle a gel. So it's called nucleoid. It's in the middle of the bacteria, and it has a different viscosity than everything else. So it is very likely that it is not only an information encoding molecule, but it is also playing a mechanical role as a charged polymer, or an electrostatic role as a charged polymer. And every transition of this molecule, which is going along with DNA, processing, may also elicit a mechanical cue. And this is something that is very exciting and which we would like to, to explore.
I think maybe in view of the time we should finish here, but there's always the opportunity to have food outside and to have further questions addressed to the speaker, and let's thank Petra Schwille again.